Okay guys, here we go, 6.3, taking a look at acid-base indicators. This is very qualitative chemistry. What we're doing is looking at things like litmus is an example of this, but indicators are these little weak acid conjugate base pairs, we'll talk more about that in Chem 30, that will change color with pH. Litmus is our first, and litmus, as we've used it before, is usually in paper form, and we've learned in previous courses that if you take litmus, such as blue litmus, which is the basified form, and you introduce it to a base, well, since it's already basic, it doesn't change color. But if you take this blue litmus and you stick it into an acid, then it will neutralize the basic form that is on the paper, tip the balance, and it will turn red as it has this dominant acidified form. Same thing happens if you take red litmus, put it in acid, it's already acidic so it stays red, but if you put it in a base it turns blue because of this. And so while you can't see this in the black and white here, when we take a look at the acidified form, we tend to see that as red. When we take a look at the basified form, notice the H is missing here, we tend to see that as blue. And then there's this kind of hybrid area where you've got both red and blue. For those of you that have taken some art or dealt with some, light blending and things like that, if you have relatively equal parts of red and blue, when they overlap, all right, you got some blue, you've got some red, when you mix those two, you get kind of a purple color. All right, and so if we actually have liquid litmus, we can actually get some very good information on approximate pHs. If litmus is definitely red or pink, all right, then it is definitely between 0 and 6 for a pH. If it is definitely blue, then it is definitely between 8 and 14 on your pH scale. If you're getting something that looks a little bit purplish, you have a transition range here, well then it is between 6 and 8. And so just given a one test with litmus, depending upon the three colors that we can get, we can start to see some very some narrowing of the pH ranges that we have. Definitely acidic, something between 6 and 8, or 8 and 14, based upon the red, purple, or blue color, respectively. Okay, we talk about red cabbage juice. It can do things because it changes colors in various pHs. Don't worry about that. All right, but we get down to something that is on our data sheet. And our data sheet lists a whole bunch of different indicators. They have some interesting names like methyl violet or benzopurpurine 48, bromocresol green. You got phenolphthalein, which we just talked, or which we'll talk about a lot. Thymolphthalein, which is its close cousin. All right, 1,3,5-trinitrobenzene. Really, really interesting names for things that are all just weak acids, which will show different colors in various pHs. So these are all really complex, usually organic molecules. We've given them little nicknames here, and you'll see that we have given them an H for their weak acid nature, just like we did for the acidic form of litmus, HLT. Okay, you can see that one right here. There's litmus again, HLT. And if we take a look at litmus again, when we are in the acidified form, you can see that it is red, which would be anything below 0 to 6. You can see that when it's in its basified form, the H is now gone. All right, it's anywhere from 8 to 14, and it looks bluish. What this shows right here, you have to remember, this is the pH range of the color change. It's the mixture of both various colors. So when I mix red and blue, I should have something purple in between. All of these various indicators therefore have three different colors. All right, if we were to take a look at, for example, orange 4, there's the acidified form. When in acidified form, it is red for HOR. When it is in its basified form, it turns yellow. So what pHs would we expect to see orange 4 be red? Well, there's orange 4. Anything from 1.4 to 0 All right, it should be in its red color. Anything 2.8 and above to 14 should be yellow. So, what would happen if we're somewhere in between? What is the mixture of having relatively equal parts of red and yellow? I think we can figure that one out. If I'm between 1.4 and 2.8, then it should be orange. Okay, we'll look at one more here. Let's take a look at bromocresol green. Okay, in its acidified form, it is yellow. HIN is the acidified form, and therefore it will be yellow in anything from 0 to 3.8. If I am above 5.4 and all the way to 14, the maximum end of our accepted pH scale, it will be blue. Well, what happens if you test a sample that is between 3.8 and 5.4? Well, what is the mixture of relatively equal parts of yellow and blue? I'll let the Jeopardy theme play in your head for a moment. And the answer is, what is green? Obviously, yellow and blue make green in the middle. So again, each one of these indicators gives 3 differing colors. All right, on the next page here, what we're going to do is we're going to apply this table. So have yours handy, whether it's from the notes or from your data sheet. And we're just going to answer some specific questions about what we might expect to see. So, here we have, the pH of a solution is 4. What color would the indicators be as we look at the data table? The first one is methyl violet, or HMV. If we take a look for methyl violet, we see it as the very first one. It is yellow at anything 0 and below. It is blue at anything 1.6 and above, and it would be a mixture of yellow and blue, or green between 0 and 1.6. If the solution had a pH of 4, then that would put it into this range, above 1.6, and it should be blue. So methyl violet would appear blue if we are in a solution that has a pH of 4. What do you think it would be if it was a pH of 11? All right, pretty easy here. Anything above 1.6 is blue, so this would not differentiate for us between 4 and 11. Both samples would appear blue. Here's another one, methyl red. All right, let's find methyl red. We go down our list. There's methyl orange. There's methyl red right there. And you can see it goes through a color change between 4.8 and 6. So where would a pH of 4 be? It would be below this, and so therefore red. Where would a pH of 11 be? It'd be above 6, and therefore yellow. So if I tested these ones, I would see 2 different colors for the 2 differing pHs. All right, there's litmus. We can take a look at this one. This is liquid litmus because it gives us a little bit more information. All right, we'll find litmus on our chart. There it is. All right, HLT. Okay, so we take a look at that one. Litmus goes through a color change between 6 and 8. So a pH of 4 would be below 6, and therefore red. A pH of 11 would be above 8, and therefore blue. Not so bad, huh? All right, here's another one, Bromothymol Blue. We have to find that one. There it is, just above litmus, HBB. All right, it changes between 6 and 7.6. So if I had a solution with a known pH of 4, I would expect it to be below 6 and therefore in the yellow range of this indicator. If I had a solution with a pH of 11, I expect it to be above 7.6 here and in the range of blue. All right, we'll do one more here for this video. We'll take a look at indigo carmine, way down here at the bottom. Okay, you can see that it changes between 11.4 and 13. So if I had a pH of 4, I'm well below this and definitely blue. If I had a pH of 11, I'm still just slightly below the color change range and blue as well. And there you go. All right, so those would be the colors I would expect in these respective pHs. We can go the other way and we can take a look at interpreting color and getting an idea of what pH range might be, and we'll do that in the second video here for acid-base indicators.